tumors. And that's for scientists and non-scientists like I really recommend this, not just for the scientific interest. There's lots of scientific interest in the monograph, but it's also a good read. I wish scientists could write like this. It used to be that scientists felt themselves to be like explorers. I mean, I still actually feel like that, but I can't express myself with that sort of sense of adventure and, here I am, and I found myself in the laboratory, and then, whether the monkeys jumped out of the trees or whatever happened. But the older science is all written in this wonderful, entertaining style. I highly recommend it. The book is also fantastic because it's amazingly prescient. Boveri, for the scientists in the audience, we know that cancer is, there are mutations that cause cancer, and there are ones that are so-called oncogenes that push a normal cell into becoming a transformed cell, and then there are so-called tumor suppressors. When you get rid of them, it's like you remove the brakes, and now the cell can kind of take off and become a malignant cell. So he was smart enough. He didn't see those genes. He didn't clone them or anything like that, but he predicted them based on observations about cancer. And the one thing that he knew that cancers had abnormal numbers of chromosomes, and he basically proposed that cancer was a disease of the chromosomes. And he specifically thought that cancer came from specific arrangements of abnormal chromosome numbers. So that's something in my own research that we've really been trying to pin down, is what is it about having abnormal numbers of chromosomes that might contribute to tumor development. Boveri, actually in that monograph, also touched on another thing which has a lot of interesting implications for the plasticity of the genome, which was that he took these, one of the ways that he messed up the division of the cells so that he could scramble the arrangement of chromosomes is that he would take these fertilized eggs and squeeze them. So when you do that, that essentially locks, the cell is trying to divide and it's kind of pinches off in the middle. That's a process known as cytokinesis. When you squeeze the eggs, actually you mess that up and they can't do that properly. And what you do is that you induce a genome doubling. So you double the number of chromosomes and you double other, I just came from a meeting called the Centrosome meeting, sponsored by the company of biologists. And you double both the number of chromosomes in these organelles in the cell called centrosomes. And Boveri postulated that genome doubling might be something that would cause cancer. And so this is I have a funny personal story about this, because my lab in 2005 published a paper where we said, I'd like to think demonstrated, that genome doublings can cause cancer using a mouse model. And we've been thinking about this. There were things in the literature. And then when I hadn't gone back actually and read, so we did this experiment. We actually had the results. And then I was writing the paper, and I was trying to, bring in a little historical, in so much as is now possible, because these journal editors won't let you say anything. I was trying to bring in a little interesting flavor of history to this thing. And I read Boveri's monograph. I think I had read it before, and then I read it again. And at the end of this thing is this description. He says, "Oh well, I think a genome doubling might cause cancer," and he knew that inflammation could trigger the cytokinesis failure and genome doublings. And he said, "Oh, and I tried this in causing an infection in the eye of a rabbit or something like this, and I could get the genome doubling, but unfortunately they didn't get cancer, and I don't know, maybe someone, will pick this up at some later date." I was sitting there reading, okay, some food. But that was fun. So that's so Boveri set up a lot of key questions, and one of the key questions is, if cancer is a disease of the chromosomes, what precisely are the changes? And what Boveri saw were abnormal numbers of whole chromosomes, and then how is it that whole chromosomes, so whole chromosomes might have thousands of genes on them, so how is it that altering the sort of dosage of the genes in the cell might do something that cause cancer? Okay, so now we can motor forward to the early 80s when I was a student. Now I'm sharing a lot of that with you. And I thought I would just, so I came into science, I thought I'd talk a little bit about how I sort of accidentally came into science and got interested in these topics. And I thought I'd touch along the way some of the advances that happened while I was a student. Because I was putting this talk together and I'm thinking, well, just sort of thinking back, wow, it's been really quite an amazing ride. And I thought that would be fun to share with you. So I started out with working with a guy at Rockefeller University who was a very famous tumor virologist named Saburo Canfuso. He won the Lasker Award for his important discoveries about cancer genes. And back in the day, we knew about genes. So genes are sequences of DNA. They get turned into another chemical called RNA, which then gets turned into another chemical substance called a protein. And that's the so-called central dogma. There's a lot more to it than that. But in for our purposes here, that'll suffice to say that the genes make stuff that is actually like the physical stuff that we are. So we knew about genes, and in principle, you could clone them and manipulate them, and you learned about this stuff in school, right? So I went to school, and I'm sure all of you had biology, and then you read that there's this thing called DNA. There are nucleotides. There are 3 nucleotides make a little code that specify, what's going to be a part of the protein, the amino acid. I don't know. So I wonder how many people here. I'm kind of a skeptic, and I don't know if anybody else felt the same way. When they taught me this stuff in biology class in high school, didn't you all think it was just a little too facile? I didn't really believe it. Did you? So, this is how I actually really got it. So I was in medical school at the time, and I was planning to be a psychiatrist. So I kind of accidentally started working in a lab because I wanted to go back to my hometown in New York City. So I started working in this lab and working on this project. We were studying these cancer genes in viruses. It was a cool environment. There were a lot of smart people around. That was fun. And then I had this idea, but the whole thing seemed completely abstract. And of course I sort of believed it, but part of me didn't really believe it because it just didn't seem very tangible. But then I thought of some little experiment where I thought, well, if I slap this onto that and do that and then put it in here, maybe that'll cause cancer. And I did, and I did what the textbook said. You put this DNA next to this one. I did it, and it worked. It was just, it completely blew me away. Which is, and I have to say honestly, it was the sort of practical realization that human beings can actually know this kind of stuff that really sucked me in. What we could do with genomes then was unbelievable. So I started out when I had to get the sequence of DNA, I'd have to do Maximin Gilbert sequencing. The graduate students know what this is. We don't have to go through it. All the people who are not scientists have to know is that this was just the most incredible pain in the ass. So it was an experiment that took several days. You had to make these finicky paper thin gels, which were always breaking. And I went into science because of the appeal of the ideas, not because of some amazing talent with my hands. And then there'd be radioactivity. It was messier to work with these things, and you do the experiment and develop the information. And so 3 billion base pairs, right, in the human genome. I mean, I'd be lucky on a good day to get 50 or 75. So I started drinking. So Rockefeller, I don't know whether this is good or bad at Rockefeller, but they have this wonderful pub. It's not quite as charming as this pub, but it's pretty charming. And they have pool tables and ping pong tables, and all the students could go, and this is terribly dangerous. The students, there was a bartender there who knew your name, and you weren't on, there was some legal rule about that you couldn't pay. Then they would have needed a liquor license or something. It couldn't have been a private club. So that means that you just put stuff on your tab, and then you got a bill at the end of the month, which is very dangerous. So I'll never forget one of the, these painstaking, long and coming experiments. I had it all set up, and I decided to go get to the bar and have a drink before I'd stop my gel and finish up the experiment. But I got involved playing pool, and I managed to run every single nucleotide of that gel. It was a sad day, but it was. I only missed 50 or 75 base pairs out of that 3 billion. So technology changed. Then there were new things that happened. Then I did some medical training. I went to MIT where I did a postdoctoral fellowship, and I was working on yeast at the time. And I lived through the most incredible revolution working on this, because in the early mid-90s, we got the first sequence of the entire yeast genome. So I had a front row seat to what that meant, because in yeast you can do all kinds of stuff. If you know the sequence, you can do a lot with it. You can take them out. It's like it's like making a collage. I mean, you can cut and paste, and you can manipulate the genome in this most remarkable way. I sort of had the pleasure of witnessing firsthand, going from this incredibly frustrating, slow, laborious thing to being able to have every gene in the genome knocked out. You go to the freezer and you want one that's knocked out for this one, that knocked out for that one. Just the power and the pace of what we could do expanded amazingly. So there's a lot of political debate now. The public, you guys, spend a lot of your tax money now sequencing genomes. And, so I think it would be fair for you to ask, are you actually getting anything for all your money? And there's a hot debate. And I think I have a good perspective on this because I'm not a professional genome sequencer. So if you would be rightfully suspicious if I was a professional genome sequencer and I came here and I gave you a talk and I said, just, send it to us and, we'll sequence more genomes and that'll be good. So I'm actually I'm sort of the collateral damage, if you will, from all this genomic information, because I've been able to live through and see it firsthand the value of having all that information coming through. So let's just say a few words about what we've learned now so we have a human genome sequence, and people have been beavering away and getting sequences of tumor genes. And there was all this skepticism about, we knew, the sort